Good evening. I'm Jill Tiefenthaler, and I'm the president of Colorado College, and it's my great pleasure to welcome you to the 2015 Harold D. and Rhoda N. Roberts Memorial Lecture in the Natural Sciences. I want to thank Professor Nina Grover for bringing Dr. Thomas Cech to campus based on their long mutual acquaintance. Thank you, Nina. Dr. Cech is Distinguished Professor in the Department of Chemistry and Biochemistry at the University of Colorado Boulder. In addition, he's the director of the BioFrontiers Institute, where his research and institutional leadership is expanding Colorado's leadership in biotechnology. In 1989, he was awarded the Nobel Prize in Chemistry, along with Sidney Altman, for the discovery of the catalytic properties of RNA. This discovery has changed our understanding of the cells and the human genome. New functions of RNA and RNA protein complexes have been discovered nearly every year since. New RNA drugs are becoming a reality. In his talk this evening, Dr. Cech will share his current research on RNA protein complexes in repairing the ends of chromosomes called telomeres. Both aging and cancer are linked to the length of telomeres. A recent article from his research laboratory in the scientific journal Nature on this topic may revolutionize how we treat cancer. In addition to his Nobel Prize, Dr. Cech was the director of the Howard Hughes Medical Institute from 2000 until 2009, the largest private funder of biomedical research. As director, Dr. Cech prioritized undergraduate education, where he instituted a major award to recognize undergraduate science teaching. I understand that many current RNA scientists, and especially women scientists, have come from his lab, more than any other RNA scholar. Dr. Cech built the Janelia Research Campus in Virginia to bring scientists from different areas together to collaborate to solve challenging problems in biology. The BioFrontiers Institute at UC Boulder has faculty from multiple fields working together and seamlessly partnering with industry under one roof. Dr. Cech's exceptional record of innovative thinking and leadership in the field is no doubt related to the fact that he is the product of a small residential liberal arts college, Grinnell College, where he received his Bachelor of Arts in Chemistry. Dr. Cech received his PhD from the University of California, Berkeley, where he studied with John Hurst, who ignited his passion for chromosome structure and function. In Dr. Cech's Nobel Laureate statement, he reflected on his time at Grinnell, observing that he enjoyed studying Homer's Odyssey, Dante's Inferno, and constitutional history as much as chemistry. He was drawn to biological chemistry because of the almost daily interplay of experimental design, observation, and interpretation. Dr. Cech is quoted on the benefits of his undergraduate experience in Fareed Zakaria's recent book, In Defense of a Liberal Arts Education. Dr. Cech compares a liberal arts education to athletics. A liberal arts education encourages scientists to improve their competitive edge by cross-training in the humanities or arts. Such academic cross-training develops the student's ability to collect and organize facts and opinions, to analyze them and weigh their value, and to articulate an argument. And it may develop these skills more effectively than writing yet another lab report. Dr. Cech's illustrious career is convincing testimony to how this preparation is the best foundation for a lifetime of transformative impact. Please join me in welcoming Dr. Thomas Cech. Thank you.
Thank you, Jill. That was a terrific introduction, and I can now skip the first several slides that I have here because you covered everything so well. As a graduate of Grinnell College, and my wife Carol, who's also here in the audience, was also a classmate of mine at Grinnell, coming to Colorado College is really special for us because we see a lot of the same kinds of students, a lot of the same values, and a lot of the same educational force of nature that these national liberal arts colleges bring. And this is the only one in the state of Colorado and one of the few in the western United States. And so it's a very special place. And it was great to talk not only to faculty but to students throughout the day. Now, I realized that the title starts with 3 letters RNA. And this is a mixed audience. There are students in the audience who have done research on ribonucleic acid, and there are others of you who probably can't quite remember what the R, N, and A stand for. So I hope you will forgive me if I start out very gently and use a little metaphor here to explain what RNA is. So you guys remember this, right? This ancient, now in museums, mechanism of information storage and transfer. Well, I choose this rather than a CD or a DVD or an iPod or whatever because of the linear nature of the information that's stored in our chromosomes is really more reminiscent of a videotape. And imagine that you have a master archival gold seal copy of your favorite film. That's sort of like the DNA. And from that DNA, you can make a limitless number of copies that are informationally equivalent. And that's like the RNA. They aren't as long-lived as the DNA, but they tell the same story. They show the same film. And then you need some kind of an output device, which for the videotape is a VCR unit. I can hardly remember what those are called. I think we still have one at home. So you plug that in, and then you see the final image on the monitor. And similar to that, the RNA, if it's a messenger RNA, gets fed through an output device, which is the ribosome, where transfer RNAs carrying individual amino acids line up on the message, and then a protein product is which is encoded by the DNA, and then the message in the RNA is the final product, just as the image here is the final product. Now, this is sort of the classical view of DNA makes RNA makes protein, but at least in Boulder, Colorado, we have a little different idea of the relative importance of these molecules. So we love this big green guy in the middle, noting that it shares with DNA the ability to carry information. In fact, some of the viruses that are the scourge of our society, like the common cold virus, the flu virus, the HIV virus, these are RNA molecules themselves. They don't even need, in the case of flu or the common cold, they don't even need DNA to do their job. So clearly RNA is a good information carrier, but what we now know is that RNA shares with protein the ability to speed up chemical reactions that are required for life. Now, why should RNA have these diverse abilities to be an enzyme and to be an active player in the body? And DNA does just this one thing, really. It just carries information. Well, the answer is very simple, that the DNA has the two strands locked in a double helix, which is very good for information storage and information transfer, sort of like a videotape. But as soon as you copy RNA from the DNA, only one of the two strands is copied. So now the RNA, being single-stranded, can find within itself regions where A pairs with U and G pairs with C, and it can form a structure which then can search out more structural possibilities in 3 dimensions and form a molecule which has a structure that depends on that sequence that was there at the beginning. So every different RNA sequence would fold up into a different structure. Now, the students in the audience know that this particular RNA molecule is called tRNA, or transfer RNA, that's involved in protein synthesis. But if the initial sequence was something different, you'd get a different RNA product. Now, how did we figure out that RNA might be able to be a biocatalyst? Well, we weren't really looking for it. We stumbled upon it. We were, when I moved to my lab in Boulder, we were looking at the gene expression in this pond animal, Tetrahymena thermophila. You'll be happy to know it's not quite as large as it looks on the screen, right? It's microscopic, sort of like a paramecium. And why would we choose this as an experimental organism? Well, because it's a eukaryote, just like human. It carries its DNA in the nucleus, and it does its protein synthesis out in the cytoplasm. But yet, these are as easy and as cheap to grow as bacteria, which do not share so much similarity with humans in terms of the way that information is stored and transferred. And so we thought this would be a simple system for understanding the process of reading out sequences from genes. But very early on, we found that this gene, like other genes in eukaryotes, did not have its sort of videotape message intact. Instead, it was interrupted by a commercial message, which technical term for this is called an intron. And, you know, you could fast forward through this, but what the cell does is it cuts it out. It actually doesn't cut it out at the DNA level. The DNA is transcribed into RNA that includes this intron. But now this necessitates another step in gene expression called RNA splicing, which is shown here, where the intron is cut out and the two parts of the message, the film that you want to really pay attention to, is continuous now without the commercial interruption. And so this was happening at a very high frequency in the gene that we were studying. And so we thought maybe we could make more of a contribution studying the RNA splicing, because no one knew the mechanism of this process. In fact, those little scissors in the cartoon were about the level of sophistication that people understood RNA splicing at the time. And we knew that RNA splicing would have to involve an enzyme, because the rate and the specificity of this process were so really exquisite that it had to be catalyzed, and it was, you could read in any biochemistry textbooks that all catalysts are protein enzymes. So we were looking specifically for the protein enzyme that would catalyze this RNA splicing reaction. Well, it turned out that the reaction just kept working whether we added any extract from the cells that would contain the protein or not. And we tried to then inactivate the postulated protein by boiling the RNA, which is very bad for proteins, but the splicing reaction resisted boiling. And then we tried adding detergents, which again are very deleterious for protein structure. And the idea now, and that didn't make any difference either, and now the idea that there was some protein enzyme catalyzing this reaction was becoming a little more suspect. And one of my first graduate students, Paula Grabowski, then gave me this for a Christmas present. She was helping work on this project. And, you know, we couldn't figure out, was there a protein involved here or not? Well, at the same time, my wife was working on transcription in her own lab near mine and lent us some RNA polymerase from E. coli. And so we were able to make a recombinant little mini gene and that had never seen the inside of a tetrahymena cell. So the idea that there'd be a contaminating tetrahymena protein was not possible. And we added her RNA polymerase. We were able to make an RNA transcript. And when that RNA spliced itself, we were ready to announce that RNA had the ability to catalyze reactions such as this. It's sort of like if, and I'm not going to go through this in detail, I went through this with the students in the class earlier today, but it's sort of as if each RNA transcript carries within it, within this intron, an enzyme, its own enzyme, so that it can cut and splice itself out. It only has to work once in order to do this job. And we then got very interested in what such an RNA enzyme might look like, because much was known about protein enzyme structure. We knew that protein enzymes were globular, they had an active site cleft, and they were, had a hydrophobic core, water-expelling core, which allowed them to fold into a globular shape. But RNA, although it was doing a very similar sort of reaction, seemed ill-suited to fold in the same way. And so in order to understand the shape of the RNA, we grew crystals of the catalytic RNA. These particular crystals were grown by Barb Golden, who's now a professor at Purdue University. And this is a 247 nucleotide RNA, about 4 times the size of the transfer RNA that we looked at before. And when we shot X-rays through these crystals from the diffraction pattern, we were able to figure out the shape of the RNA. And several other laboratories also were making contributions in this area. 2004 was a big year when 3 different self-splicing so-called ribozymes, catalytic RNA molecules, structures were solved. They all had the same sort of core at the middle, the same sort of active center, but it was sort of buttressed and held together by different elaborations of RNAs on the outsides, which makes these 3 structures look rather different. So this helped establish the idea that RNA, although chemically very different from protein, can end up folding into specific 3-dimensional structures to catalyze reactions. And I'm going to skip the next one. So what were the implications of this? Well, the first one had to do with not Campbell's primordial soup, but perhaps the origins of life on the planet Earth. Scientists have always been very interested in the origins of life. If very simple self-reproducing chemical entities occur by random chemical processes, then what would such a very primitive self-reproducing system look like? And you look to sort of current biology to come up with an answer. And what do you see? You see that, well, you need an informational molecule. If you don't have information, you can't inherit progress that you make from one generation to the next. I know it doesn't seem like we're making much progress, but over time, over millions of years, we've made progress, right? So you need some kind of a nucleic acid molecule, perhaps, to be the informational molecule. But nucleic acids, namely DNA, were thought to be inert. They couldn't copy themselves. They had to wait around for a protein enzyme to come along and duplicate them so that you could go from one generation, from a mother generation to daughter generation. So now that we know that RNA can do both things, it can be both an informational molecule and a biocatalyst, maybe at the beginning there was RNA, and then the proteins came along later, and eventually the DNA as a more stable storage form of genetic information. Well, whether this so-called RNA world hypothesis is historically accurate or not is difficult to be sure of. But one thing we know, that whether or not these really catalytic RNAs evolved into RNA protein complexes and eventually into our modern proteins, we do know that the last steps of this are still within us now, because many of the catalysts within our own bodies are not just protein enzymes, but they are RNA protein complexes. So the next part of the title of my talk is this telomerase enzyme, and I need to introduce that by, again, showing you a cartoon of how it is thought to work. Now, at the very ends of all of our chromosomes, or the chromosomes of other eukaryotes that have linear chromosomes rather than circles, the DNA cannot be copied or replicated by the enzymes that are really good at replicating the vast middle stretches of the chromosome. Instead, at the very end, there's sort of no Watson strand to template the Crick strand, or no Crick strand to template the Watson strand. So out at the very end, there's this entity discovered by Liz Blackburn and Carol Greider called telomerase, which carries around with it an RNA molecule, and that RNA serves as the template for extension of the DNA. It extends it by 6 nucleotides at a time, and then it ratchets back and it builds it out again and ratchets back. But sadly, this RNA, this telomerase RNA, was not a ribozyme. It was not able to do this all by itself. It had to work as an RNA protein complex. And people were searching for this yellow egg for about 10 years, and then Joachim Lingner, a brilliant Swiss biochemist in my lab in Boulder, eventually purified it and found the protein part, and we were then able to put it all together. The protein plus the RNA made the active unit. I'm just going to have 2 slides about how Lingner discovered this, because I think the students might find it interesting. So everyone else, there were some dozens of people around the world racing to try to find this protein. Everybody else was looking in human cancer cells for reasons which will become apparent in the next part of the talk, because there's a strong importance of this telomerase for cancer. But we instead, in our lab, were looking at another one of these ciliated protozoa called Euplotes aediculatus. Well, why would we choose a weird organism like this? Why not cancer cells? Can't you get more money to work on cancer cells for research funding? Well, each of you out there you look like, to me, like you're mostly diploid organisms, okay? So in each of your cells, you have 23 chromosomes, haploid, times 2, 46. Each one has 2 ends. Let's just round that off and say you each have about 100 telomeres in every one of your cells. Well, this strange creature has 100 million per cell. And each of these tiny chromosomes is capped off by canonical telomeres and has to be replicated by telomerase. So I sold to Lingner the idea that use this organism, you'll have a millionfold head start on everybody else. Well, he bought into that, and then he found out it was about 1 million times harder to grow these cells than any other cells. In fact, we grew them on Pyrex lasagna dishes that we got from King Soopers. And they grow with bacteria and algae mixed in with them, which you then have to separate out later on. So it wasn't the best system, but nonetheless, he persevered and was able to purify telomerase from Euplotes. And then, since nature is the grand recycler and never invents something just once because we're all related, it turned out that by computer database searching of the human genome, we were able to find the entire family of these telomerase reverse transcriptase proteins, and the human one was among those, okay? And why do you maybe needs just a second to talk about why is it a reverse transcriptase. Well, if you think about what HIV reverse transcriptase does, it converts an RNA virus into a DNA copy, which then gets cemented into one of our chromosomes, much to our detriment. But what does telomerase do? Well, it also copies RNA into DNA. So again, nature, the great recycler, there's an evolutionary connection between this viral HIV, retroviral reverse transcriptase, and the healthy telomerase, which is extending the telomeres in all of your cells. So TERT stands for telomerase reverse transcriptase. Okay, so what did I do? I got a drink of water and everything went blank. But I can still see it on my laptop. So let me continue. Oh, thank you. So there had been a story going around in the scientific community that maybe the aging of human cells was tied into the lengths of the telomeres. The idea was that about the time you're born, most cells, except for some stem cells, quit making telomerase. The telomeres are no longer extended, and then the failure to replicate them ends up with the ends of the chromosome shrinking with each cell division. And when they get to a critically short length, a signal is sent to the cell to quit dividing. And this is thought to be part of aging, that the failure to extend telomeres causes the cell to age and then to quit dividing. We call this cellular senescence. Now, once TERT was discovered, a number of laboratories, most famously a laboratory in Texas, reintroduced the gene for TERT back into senescent human cells and was able to immortalize them. That's a pretty cool experiment. And that got a lot of people interested in the possibility of lifespan extension by adding telomerase. In fact, one of the companies working on this said they kept getting phone calls asking, how big is the pill going to be? I want to take that pill, okay? And there's an interesting book that's been written about the discovery of telomerase and it just, this is so interesting. It just disappeared. Well, it's called Merchants of Immortality, and it's talking about how hyped this whole idea was of becoming immortal by having more telomerase activity. Because you see, the age of our individual cells is not necessarily exactly the same as the age of the human organism. And if we could stop the aging of individual cells, for example, stem cells, there are many human diseases that might be treated by rejuvenating the cells in your body that need to keep dividing. But if we had all of the cells in our body not knowing when it was time to stop dividing, we would be very large and probably a very large tumor for reasons that you will see in a minute. And so although my parents were amongst the ones who were asking me, when is the therapy going to be available, the truth is that there's no direct correspondence between this cellular aging and the aging of the human body, which is influenced, for example, a lot by the fact that we accumulate oxidative damage in our cells as we grow older, both in our genomes and in our proteins, and that's hard to erase. So telomerase then in health and disease is a very delicate balance. If we have too little of it, especially in our adult stem cells, they quit dividing and they age prematurely. And there are quite a number of human diseases that can be traced directly to a failure of there being enough telomerase in the bone marrow, in the lungs, in other tissues. On the other hand, too much telomerase isn't a good idea either, because cancer, which has to be immortal, continues to grow, occurs in part through reactivation of the dormant telomerase, turning on this gene for the TERT protein again. Okay, so if we have too much, that also is unhealthy. Now, this has been known for quite a time, but yet it seemed like this TERT gene, telomerase reverse transcriptase, was a strange cancer-causing gene, or oncogene, because the other oncogenes that had been discovered either had mutations in them that activated them, or some kind of gene rearrangement. You may have heard about the Philadelphia chromosome, BCR-ABL, kinase being rearranged. That kind of thing is really common for cancer-causing genes, and TERT didn't have any of these. But then, 2 years ago, February of 2013, 2 laboratories, one at the Dana-Farber Cancer Center in Boston and another at the German Cancer Center in Heidelberg, reported that everybody had been looking for mutations in the wrong place. They had been looking within the gene body itself, and instead there were recurrent mutations that were specific to cancer in the gene regulatory region, upstream from where the actual protein coding region is located. You didn't even have to look very far. They were only 120, the most common ones, only 124 base pairs away from the place that the first codon for the TERT protein. So they were in the so-called promoter for the gene. And the reason that people didn't look there was just because the technology, up until a few years ago, it was expensive to sequence whole genomes, and so people were just sequencing the protein coding portions. Now, how could one base pair mutation change a cell into immortality? Sounds pretty science fiction-y. Well, their prediction was that this one base pair change created a new binding site for a particular kind of transcription factor, abbreviated as an ETS transcription factor, a protein that would sit there and would encourage this gene to fire off, and that was the key for turning on TERT and thereby making cancer cells immortal. Now, this was originally found in melanoma, and remarkably, 80% of melanoma has this same base pair change, and the adjacent normal healthy skin tissue never has it, or rarely has it. Okay, so it's really tumor specific. But then this same base pair change started being found in brain cancer, in glioblastoma, liposarcomas, bladder cancer, liver cancer, which is a huge killer, and a number of other cancers, but interestingly not found in prostate cancer or breast cancer. So about half of the common cancers have quite a remarkable frequency of this change in the TERT promoter. So this is what we call an association, right? It doesn't prove that this is causal, but when you see something associated that often, it is sort of a major hint that something is going on. So therefore, someone from the Journal of the National Cancer Institute wrote this review about these findings and said that this TERT promoter is eyed with suspicion, but also with hope, right? It was sort of too good to be true. And I love the woman's last name who wrote this article. Her name was Delude. So were we being deluded? So this discovery led to new questions which we've been able to answer in the last couple of years. So first of all, is the TERT promoter mutation simply one of many pathways to reactivate TERT, or is it associated with particularly good at activating it? What are the clinical implications? We're starting to understand these. Okay, there's this association between the promoter mutation and telomerase, but can we establish that the mutation really causes the gene to be expressed more? And finally, can we test the model that came from these two papers I just talked about, that these promoter mutations gave about a 2 times boost of the amount of the telomerase that was being made? Always seemed a little strange to me that a factor of 2 would be such a big deal in biology, but often it is. So a group of students in my lab, including Natasha Powell, who was a freshman at the time from the Twin Cities, and Sumit Bora, who's a postdoctoral fellow, came from Yale, collaborated with the people at the Cancer Center at Anschutz in Denver and got a bunch of bladder cancer cell lines. The reason for choosing bladder cancer was because our colleague Dan Theodorescu in Denver is a bladder cancer expert. We could have chosen melanoma or brain cancer or any of these other ones. And they looked through they sequenced the promoter region. It's a pretty cool project for a freshman in college. And what Natasha found was that about 2/3 of the cell lines had the advertised -124 mutation shown in red. Most of them were heterozygous for this. In other words, you have 2 genes, right, one from Papa and one from Mama, and that the one of the genes had the mutation, the other one didn't. Only in one case did she find homozygous mutation, and then we had a bunch of cell lines that did not have the mutation, which we could use for comparison. And we developed enzyme assays to look at the amount of the activity of the telomerase in these bladder cancer cell lines. And this is an assay where we add we pull out the telomerase from the cell using some pretty fancy tricks, and then it's an antibody purification. And then we add an artificial chromosome end and see if the telomerase that we've pulled out of the cell is able to extend that as sort of a surrogate of what would happen in a living cell. And you can see it adding 6 nucleotides, 12, 18, 24, et cetera, to this artificial chromosome end. And all that you can really see from this particular experiment is that there's a huge variation between different cancer cell lines. Some have high levels of telomerase activity, some much lower, some hardly detectable. So that was one of the sort of unexpected findings from this work, is that there was this enormous variation, so each of these dots is the amount of the TERT messenger RNA in the cell line, the amount of the TERT protein, and you can see that they both the ones with the wild type promoter and the -124 mutant promoter have a big range, but that the median values are very different between the two. Good correlation between the amount of messenger RNA being made and the amount of protein, sort of a linear relationship. And then if we look at the telomerase activity, again, the ones with the mutation making about 2 times more than the ones without the mutation, and again, a pretty good relationship between the activity and the amount of message, or the activity and the amount of protein. The RNA subunit of telomerase is already there in abundance, even in normal human cells, for reasons that we don't fully understand. And so therefore, since it's not limiting, there was no correlation between the amount of the RNA subunit and the amount of telomerase activity. And then these different cancer cell lines also had really long chromosome ends in some cases, shown here, and really short ones in other cases. Big variation, but again, the ones that had the mutant promoter, on average, had more length to their telomeres. So what does this have to do with the disease itself? So we were able to reanalyze 2 patient studies, one that was done in New York City at Memorial Sloan Kettering, and another one done at an Asian hospital, in which they had not only followed patient survival, this is called a Kaplan-Meier curve, so this is disease-specific survival, the number of people who didn't die because of bladder cancer as a function of time in months after they had cystectomy to remove the primary tumor. And then these same patients had all of their RNAs in their tumor cells analyzed. Well, in this vast database, the TERT messenger RNA was included, so we were able to look at the ones that had the high telomerase expression, shown in green, and the low telomerase expression. And you can see, you really don't want to be one of these cancer patients that has the high telomerase. This is not good news. And therefore, something that we need to work on is trying to inhibit this enzyme to try to revert this bad prognosis, this green curve, back up to this somewhat better prognosis, the blue curve, or perhaps even flatten that out. Now again, all of this is correlative. Is this really causation? All I've shown you is association and more association. So there's a new technology, which perhaps some of you have read about, called CRISPR-Cas9 genome editing. This is a bacterial defense system, prevents bacteria from getting infected by bacterial viruses, and remarkably, if you move its key components into a human cell, or into a mouse, or into a fruit fly, into any of those organisms, it works really efficiently and is able to change the genome in a predetermined way. Tremendous research opportunity. So what we've been able to do is to use this CRISPR-Cas9 system in the cancer cells to cut at the beginning of the TERT promoter and introduce a gene for a jellyfish gene for a green fluorescent protein. So now the cells that have successfully undergone this insertion glow green in the dark. We can use an automated cell sorter to collect all of the green glowing cells, but you notice that my student, Linghe Shi, who did this experiment, also inserted a mutation in the promoter right next to the green fluorescent protein. So now these cells have not just the green fluorescent protein, but also a single base pair change at this one gene out of 20,000 in the human genome, and now she can use a second round of CRISPR editing to cut here, leaving the mutation in, cutting out the green fluorescent protein, now sorting cells for dark cells, ones that are not glowing green in the dark. And when she sequences the winners at the end, she finds that she has transformed the original promoter region to one that has a single base pair change. And we can use this genome editing to make, for example, cell lines that have, and here's the critical position, this -124 position. Here you can see a clean DNA sequence. Both mama gene and papa gene have the same sequence. You get to the -124 position, heterozygous at that region, then the sequence is clean again. We can take those cells and we can convert them either to completely mutant or completely wild type. You can see now there's only a red nucleotide at this sequence. Now there's only a blue one instead of there being equal amounts of red and blue. And lo and behold, the expression of the TERT gene is increased when we have 2 mutant promoters and decreased when we have only 1 mutant promoter. We're not sure yet why this didn't go down to 0, but it did go down quite a bit. And these experiments are still in progress. This is unpublished data of Josh Stern in the lab, but it looks like the mutation is causal, not just associated. Okay. Now, for the last part of the talk, I'm going to get into the area of epigenetics. And I thought, again, this was a term that many of you would be unfamiliar with, and so that a few slides of introduction would be appropriate. Now, the way that normal genetics works is that you're sort of like in a dynamic equilibrium between mom's genes and dad's genes, right? You have an equal chance of inheriting a version of the gene from either of your parents. But there are some strange exceptions to this called epigenetic inheritance. And in the next slide, this particular one is shown as a mouse genetic cross experiment, where the genetics seem very strange. This is paternal imprinting in this particular kind of epigenetics that I'm choosing as an example. So you've got dad has the green gene. I've actually never seen a green and a blue mouse. But anyway, this is just for illustrative purposes. I pulled this off of Google Images. Dad has the green gene, mom has the blue gene, even though the kids both have an allele from both parents, the one from mom doesn't count. And you only express the one from dad, so all the offspring are green. And conversely, there's something called, that would be paternal imprinting. There's also maternal imprinting where, I'm sorry, this is still paternal, okay? So now it's the other way around. Dad's got the blue gene, and in all of the offspring, even though they inherited genes from both parents, they're still paternally imprinted, and so they're all blue. Okay, so this epigenetics comes from the Greek. It's something sort of superimposed on top of normal genetics. It's heritable changes in gene expression that are not attributable to nucleotide sequence variation, which is the kind that we are more accustomed to studying. Well, how is this possible that it doesn't matter what the sequence of the gene is, it can be just not even expressed. It's as if it weren't even in your cells. Well, what happens with epigenetics is that even though the A's, G's, C's, and T's on the DNA are unchanged, either the DNA itself is chemically modified by methylation on cytosine residues, or the histone proteins that wrap up the DNA into nucleosomes are modified on their tails in such a way that the portion of the chromosome containing this gene condenses into almost a crystalline array of chromatin where transcription is impeded. So instead of being in an open state where it can be expressed, it's in a closed state. So that's how you get epigenetic silencing of genes. And we found that this is we weren't looking for it again, but we found this is what's happening at the TERT promoter. That there was this, but now it's happening because of this one base pair change at position -124, not because of the parent that gave the gene. So there's this dramatic allele specificity that the promoter with the mutation has the RNA polymerase, which is going to copy the gene, associated with it. So here's what the DNA, if we look at the DNA itself, we see at position -124, we have both the mutant copy and the wild type copy. So unlike the other positions that give a clean DNA sequence, we get a 50-50 sequence at this position. Now we look at which gene is associated with RNA polymerase II, and it's mostly the red copy, the mutant one, very little of the blue allele. Now we look at which gene is epigenetically silenced by having its chromatin modified. It's the other one. The blue one is mostly silenced. The red one is not associated with this histone 3 methylation mark. And this was in liver cancer cell lines, but we've done the same thing here in another liver cancer cell line. Again, we see that the DNA does not look like what is associated with these various proteins. And if we look at where the messenger RNA is expressed, we see that it's expressed, in this case, we have to look at, remember, the promoter region of the gene is not the part that's transcribed. It doesn't make RNA. So now we have to find a marker fortuitously within the transcribed region of the gene that's only there in one of the two copies. And this is one of these so-called SNPs, single nucleotide polymorphisms. You see the two copies in the DNA. You look at the RNA that's being made, and it just has the green nucleotide at this position. And that's the one that upstream from it has the mutation in the promoter. And I have more examples of this, but we don't need to look at more examples. Let's just look at the model that explains all of this. Okay, so that we think that the TERT gene in most of our cells in our body that are not cancerous is in this epigenetically silenced state. The chromatin is condensed, you're not making any messenger RNA, therefore you got no TERT protein, you've got no telomerase activity, the telomeres are shrinking. Cell is aging, which is a good thing, right? Because the alternative is cancer, okay? So now this mutation, just by chance, happens. It's a rare event, but one cancer cell out of 1 million happens to get a mutation in the TERT promoter. That attracts this ETS family transcription factor, which then attracts the machinery that changes the chromatin from being stop signs, condensed and inactive, to being a go sign, extended and transcriptionally active. It recruits RNA polymerase and makes the message, which then causes this, so then this epigenetic switch that makes the message causes more TERT protein, more telomerase activity, allows the cancer cell to have its telomeres maintained. Those cells become immortal, and pretty soon the tumor, which is also sort of a Darwinian experiment, is just 100% cells that have this TERT promoter mutation. The other ones just can't keep up. They die off, and now the tumor is enriched to almost homogeneity in cells that have this particular mutation. Then this leads to poor patient prognosis, at least in bladder cancer. But remember that this is one of the most frequent mutations in all of cancer, so these results may be much more broadly applicable than just in bladder cancer. So, okay, we can understand it. What can we do in the future? Well, the idea right now already, cancer patients are very often having the genome of their tumor sequenced, right? You don't care about your genome as much as you care about the mutations that are present in the tumor that is killing you, and that if this tumor genome sequence identifies a TERT promoter mutation, then we hope that in the future there will be anti-telomerase therapeutics which will shut this down. And in closing, I would like to thank the students and postdoctoral fellows in my own laboratory, our collaborators in Denver, in Australia, at Johns Hopkins, in Connecticut, and also the funding agencies that support our work. And I would like to thank you for your attention. And I'm happy to answer some questions. I know the last part of the talk was quite technical, but the students here at the college are very bright and have had a lot of this in classes, and so I wanted to give them some very recent information, even if it's not completely finished and not published yet, and we're still grappling with it. And I can't see if anyone's raising their hand because of the spotlights. Okay.
Hi. Oh, wow, microphone's special. You were talking about the undergraduate who had been looking for the mutations, and the vast majority of them were heterozygous instead of homozygous. And then when you had in vitro experiments, you had both of those cases. So is there an easy explanation why it would be, is it what you were talking about, it's just a race in a Darwinian experiment and you only need one copy and you don't have time to get both? Or?
Well, so again, we have an explanation which we think is reasonable, but that doesn't mean it's correct, okay? So the question, if you all heard it, was how come, you know, if in our artificial experiments 2 mutations gives even more activity than 1, how come these tumors tend to have just one of the 2 genes mutated? And our explanation is that 1 is probably enough to give cellular immortality. So the sort of incentive to have a second mutation is gone. Once you have the 1, that tends to be, in most cells, enough to give enough telomerase to make the cell immortal. But that's a testable idea, and I'm open to the possibility that it's wrong. And your other thing you mentioned is maybe there just wasn't enough time for a second mutation. That might be another possibility. Yes, in the... Could you speak in the microphone? So that...
Do you imagine that germline TERT mutations are not viable?
You know, so the question is, what if you inherit this mutation instead of getting it de novo?
Or de novo, but germline.
Right, but germline. And there has been one paper on a family that was passing this mutation around, and they are incredibly prone to cancer at a very early age, and they are getting a wide variety of cancers. It's not just bladder cancer. They're getting this whole range of cancers. So the idea is, so why wouldn't it be, I would have expected what you said, that it would be embryonic lethal, but maybe they have something else in their genome that suppresses this to some extent so that they are born normally, they grow normally, but they're extremely cancer prone. Good question.
Sorry, I have a question. To what extent does this co-occur with mutations in tumor suppressor genes that are known to have chromatin remodeling abilities? Like, I know that BRCA1 can cause the methylation or demethylation of particular histones. Is that those related?
Yes, well, so it almost always, I hope I didn't give the impression that this was sufficient to give cancer. There are also other oncogenes that have to be activated of a wide variety of sorts that differ from cancer to cancer, and also tumor suppressor genes have to be inactivated, as well as the TERT being turned on. In order, minimally, those 3 things have to happen. And then for metastasis, probably still other mutations. So your question, I think, was whether there were particular other cancer mutations that co-segregated with this one. And so far, it doesn't seem that way. It seems like this is sort of superimposed on the other array of already known cancer mutations. It would be really exciting if there was some particular chromatin remodeling oncogene that, you know, was either mutually exclusive with this or always co-occurred with it, but that hasn't been demonstrated yet.
You mentioned the
anti-telomerase therapeutics possibly in the future. How much work has been done towards that, and how feasible is it, I guess, as an actual target?
Right. So,
Do you think?
so there was a lot of work done after the discovery of TERT. In fact, the University of Colorado licensed it to a number of companies who tried to develop pharmaceuticals. Nothing on the market yet. There's still one drug in clinical trials. But more recently, there have been some people saying, you know, that's very slow progress. We need to think outside the box. And instead of trying to develop a normal enzyme inhibitor, maybe we can inhibit other steps in this pathway more effectively. And so our own lab is working on a drug that we're trying to find a drug that would prevent the telomerase from latching onto the telomere where it has to work, rather than being a traditional enzyme inhibitor. Is that a better idea? I'm not sure, but it's a different idea. So it gives us a fresh start on the problem. But
Could
it's
this have negative effects in other cells?
So the question is, what would be the side effects? And yes, it would be bad for stem cell biology in general. So it would be bad for your gut epithelium, for your bone marrow, but the hope is that in many cases, cancer cells, for whatever reason, have shorter telomeres. I showed you that isn't always true. But in some cases, cancer cells have shorter telomeres to start with than the normal human cells. So they are one step closer to being inhibited by a telomerase drug. So in those cancers that carry, where the tumor has short chromosome ends, we may be able to kill the tumor cell before there would be much effect on stem cells in your normal tissues. Yes?
I was just wondering if the...
Would you mind using the
okay.
microphone?
I'm sorry. I was just wondering if your, the treatments that they are using now for cancer, like chemo, if that is affecting these these cells, or the growth of them, is it negative, is it positive? Have you done any studying on that?
We have not done any studies. It's a very interesting question you asked, though. So let me just rephrase your question. Question is, would the high telomerase cells or the low telomerase cells be more susceptible to the sort of standard chemotherapy? And one could imagine that there might be a difference, but it's not been well enough studied yet to be able to answer that question. Yeah, go ahead. You're holding the microphone. Go for it.
You mentioned that for some diseases, if you could encourage the production of TERT, it would help, like degenerative diseases, that would
Yes.
be a productive way to perhaps combat that. And in some cancers, it would be productive to stop TERT production.
Yes.
Would those two paths of research be similar, or would they require completely different approaches?
No, thank you for the question. It's a great question. You know, I think that in the drug discovery world, people talk about antagonists, things that shut something down, and they talk about agonists, things that encourage a process. And so in the process of looking for molecules that would interact with TERT, one might, if you set up the assay in the right way, you might find small molecules that bind in a way that makes it more active. And that would be perfect for these diseases of telomerase insufficiency, because most of those patients are only sort of like, they're only 25% too low or 50% too low. They're not zero. And so if you could boost up their activity by a factor of 2, you'd be back to the normal level. And if you could titrate it just right, you should be able to do this without making them now cancer prone, right? You don't want to overshoot. You want to just get it back up to the normal level. So I think there's quite a bit of future hope, and particularly a lot of people with aplastic anemia and pulmonary fibrosis. Those two diseases have a lot of causation from telomerase. Not always, but there's a big fraction of those that have too little telomerase. So that's a good question.
Follow-up, if someone had one of those two diseases and cancer,
yeah. Well, so this again it turns out that that's been well studied, and I'm sorry, the answer is going to be slightly long. Some of you will run for the door, but it turns out that you can also be predisposed to cancer if your telomeres, if you fail to elongate telomeres. You say, well, why would that be? It should go there. I just have been telling you it's when you have too much, right? When you reactivate. Turns out, if the telomeres get too short, you can get genome instability, and especially if you have like a p53 mutation, so that these cells don't kill themselves. A lot of cells in the human body are smart enough, when they're really damaged, they kill themselves, and we never know it, because just missing a cell, it doesn't hurt us, right? We've got lots of those. But if they have their tumor suppressor activity mutated, and now they get super short telomeres, they undergo a lot of genome rearrangement, which leads to activation of other oncogenes. So surprisingly, some of the families that carry these diseases of too little telomerase are also extremely cancer prone. That's why I say it has to be, it's right on a knife edge. You don't want 2 times too much, and you don't want 2 times too little. You just have to be, it's like Goldilocks, it has to be just right. And so that could be that is an issue with the people with too little, that they can get cancer too. Yes, in the back, if you could hand her the mic.
So there are a lot of cancers that it doesn't affect, and there are a lot that it does. Of the ones that it does affect, is there any correlation in the DNA that makes them more susceptible?
Well, this is you're asking a question which was like last week's phone conversation between me and a bunch of cancer researchers. So there are 2 theories on this. Like one would be like with the breast cancer, the prostate cancer. They don't have the mutation, but they still upregulate TERT. And so one possibility is that their mutation is not so easy to find. Instead of it being right near the beginning of the gene, it's 1,000 miles away, and because the chromosome loops around, it comes into proximity. So that's one theory, and some of our colleagues at Johns Hopkins don't like that. They say, we've looked really hard for that mutation in breast cancer. We don't think there is another far away one. Well, but then what's the other competing theory for upregulation of telomerase in breast cancer? And there are some other possibilities, but none of us like, it's sort of like we have several possible explanations and they all seem rather unlikely. So right now we just have to keep thinking about it. Yes, microphone down here. Oh, up there first, please.
Oh, I have a question. What about a non-cancerous state like progeria and this particular gene? Has that been studied, or what do you know about that?
Progeria, which is a disease of early, very vastly premature aging, has been studied because telomerase would have been one of the candidate genes. And I don't believe there have been many or any cases of progeria that have been linked to telomerase mutations, for example.
Okay.
But it was a good question, but it just doesn't turn out to be the case.
So I have a question about the ETS transcription factors. How
Yes.
specific is the binding? Is there any promiscuity such that in some of these other cancers that don't actually have the promoter mutation, you might actually be getting perhaps even an ETS mutation that allows it to bind in the open reading?
So the question is how much of the ETs do you need to have in the cell, or?
No, just is ETS very specific in its binding, or is there any promiscuity such that it can bind some of, if there's a SNP other than the one that you're looking at? So you're not necessarily having the same exact SNP, but ETS might be a little bit more promiscuous, so you're getting upregulation of telomerase.
Actually, I think that's a great theory. I'll add that to my list. And the reason I think that might be a great theory is that often these transcription factors collaborate, and if they bind near each other, then they can help each other bind. And so your idea that maybe that maybe we're looking for too specific of a site, and maybe the mutation is rather different, but still ends up recruiting it, could certainly be true if right next door there was a helper protein already bound there. Now you might not need to have a perfect mutated site to attract the ETS transcription factor. But you have to realize, now that you've made that theory, that's much harder to look for, right? Because you use the word promiscuity, and now your computer search is going to have to be promiscuous, right? And that, for a computer programmer, they don't like a lot of ambiguity. They want to look for a specific thing. So I'm not saying that will make it impossible for us to find it, but it would make it tougher. But it's a good idea.
So I just had a question about sort of the broader macro picture of cancer research. There are obviously so many associated factors for cancer. There's a huge variety of oncogenes. Like you said, not all cancers even have these TERT promoter mutations. Of course, there are tumor suppressor mutations. So is it a sort of scenario in which you can develop something like a telomerase inhibitor and break one step in a chain of many things that have to go right for the cancer to occur? Or is it a situation where you have so many associated factors that you're sort of playing whack-a-mole with your cell's biochemistry?
I'm somewhat optimistic. For many years we've been told, there's going to be no silver bullet for all of cancer. It's too complicated of a disease. Every cancer is a different disease. But when we do this CRISPR gene editing and change that promoter, the guy who's culturing the cells tells me, and this is just from last week, says that the ones with the 2 mutant promoters are growing like gangbusters, and then the ones with the 2 wild-type promoters are hardly growing. Now, this is in plastic petri dishes, right? That's not the same as a tumor in your body. But it sure makes me optimistic that maybe this could be very propitious for a lot of cancer patients to interfere with this pathway. So it's the thing you said, even though you've got all of these steps, if you knock out a critical step, then the cancer may have trouble continuing to proliferate.
With mercury being as damaging as it is and so many of us having it in our mouths, I was wondering if...
Where is the...
I'm right
okay,
here.
yes.
If you think that might have an effect.
If mercury might have an effect on...
The damaging...
Yeah, hasn't been studied. In general, heavy metals can have deleterious effects on a lot of DNA processes. So I wouldn't poo-poo the idea that could have an effect. Yes.
Or would it cause the tumor to shrink? Should I repeat that?
Yes, so the question is, if we could inhibit the telomerase, would it really cause the tumor to shrink away, or would it simply stop its growth? And my guess would be that it would be more like stopping its growth. Although now with all of these new immune activating drugs being developed by a number of pharmaceutical companies. I know about the Merck one most because of my relationship with Merck, but Bristol Myers Squibb, a number of other companies are also developing ways to stimulate the human immune system, I would imagine that maybe if you couple that with something that stops the tumor from growing and now stimulate the immune system, it may be able to clean it up. But so these combination therapies are, just as in AIDS, AIDS was really difficult to conquer with any one drug, and it's really when they got to the 3-drug therapy that they could really make a huge impact on the lives of AIDS patients. We're thinking that in the future of cancer research, the same thing is going to be true, that instead of giving cancer therapies one at a time that hit a tumor with 2 or 3 drugs simultaneously, and maybe the escape from that will be difficult for a tumor.
Follow-up question, which is, do you think it would help with satellite colonies and stuff? Like, would it be optimal to hit it really early before it spread?
I think always, yes. I think a lot of the success we've had with cancer is earlier detection.
I just had a question. I was thinking you talked a lot about trying to inhibit the enzyme activity of TERT, of the protein. Has anyone looked into trying to inhibit the RNA component of telomerase? Can you use an RNA therapy, bind up the RNA and cause some sort of a problem? I know you said the RNA levels sort of stay the same, that it's really the protein that gets...
No, but your point is still good, because even though the RNA levels aren't limiting in the cancer, if you knock them down to 0, they would become limiting.
They would become limiting,
because
and
it
then
has to be
TERT would be off, essentially.
Right, so, exactly. And it turns out that the one drug that is in clinical trials right now is an RNA inhibitor.
Or a sponge, right? You could come up with, you could create an RNA sponge where you're just sucking up.
You could create a sponge. This particular one, I mean, sponges work well in the research lab. It's a little hard to do in somebody's body because you just have to express so much of something.
Right.
But what this little drug is a little like it fools the enzyme into think it's binding a telomere, and instead it's a piece of chemically decorated nucleic acid,
kind of an
which
LNA.
covers up that template region.
Yeah.
And again, it works well as a research tool. In the clinical trials, it remains to be seen.
I also just have sort of an odd question, probably, but you said you did an IP with TERT. Does it bind anything else besides telomeres? Do you see it binding any other kind of RNA? I guess I'm coming from an RNA biologist standpoint. I always think these things always seem to have a dual function. It's rare that you have an RNA binding protein that just does this one thing.
Yes, so this is a very controversial part of the field. It's called, non-telomeric functions of TERT. There are whole review articles written about these, and it turns out that in a number of our laboratories, when we've tried to reproduce these findings, we have sort of shown that they aren't true. And then I go to a conference and I talk to somebody from Berkeley, and he's really angry about this because he spent 2 years trying to follow up on one of these stories, and he also, for a completely different reason, thinks that it's untrue. So so just because the published stories are hard to build on doesn't mean the concept is wrong, right? So it's an interesting idea, but I would caution you, if you read any review articles on this, don't believe everything you read. So I don't think good examples of non-telomere functions of TERT have been found yet. Should we do one more question and then excuse the class? Or 2 more questions, because there's a fellow down here who's been, yes, where are we over here?
Right here.
Okay, good.
So I was just wondering if instead of trying to stop the mutation in the TERT protein, if you could just stop its association with the telomerase, would that have any sort of effect on its cancerous activity?
I think so. I think that's whenever you have one of these complex assembly pathways, it gives you the opportunity to intervene at any of those steps, right? It's like a linear path. You cut the road at any place and you've solved the problem. People tend to look for protein enzyme inhibitors because that's what drug companies have done historically for many years. But that doesn't mean that these other layers couldn't be potentially better places to intervene. Let me take them, I'll take the microphone down to the, one more fella down here. Oh, you have it already, okay.
As far as the 2-point mutations go, can you eliminate any type of gene rearrangement or transposable element as the cause of that?
We looked, it does happen rarely, and there are sometimes, especially in liver cancer, there can be a hepatitis B insertion near TERT that is driving the TERT gene. But that turns out to be, that was a rare event. And whereas this mutation is not usually accompanied by any other rearrangement.
Thank you.
Thanks, those were terrific questions. Let's give a round of applause to the audience. Yeah.